students, welcome to Biomix. Do you know where does a plant synthesizes its food? It is in the leaf. So if it is synthesized in the leaf, how it is being transferred to different parts of the plant? How does a root get its food? So for understanding that, we have to understand this topic, that is a phloem loading and unloading, in which we will discuss how the food is being synthesized in one part of the plant and it is being transferred to different part of the plant. The part of the plant where the food is being synthesized, it is known as a source, and where it is being transferred, it is known as the sink. So what happens in case of a vascular plant? There are two things, that is the xylem and phloem. This xylem which is present, it is helping in the transfer of water, and the phloem which is present, it is helping in the transfer of food material, that means transfer of sucrose. Okay, why the sucrose is being transferred? Because the sucrose which is present, it is a non-reducing sugar. So how the food is being formed? This food which is present, it is being synthesized in the form of glucose by the plants by the process of photosynthesis. So this phloem which is present, what is this phloem? This is a complex, it is a complex parenchymatous tissue. So what do you mean by complex parenchymatous tissue? That means it is made up of many different things. It is made up of a complex process. The first thing which this phloem is made is the phloem fiber. And this phloem fiber, what it is? It is a dead tissue. Why it is a dead tissue? Because this phloem fiber which is present, it is made up of sclerenchymatous tissue, which is a dead tissue. The second thing which is present is the phloem parenchyma. And this phloem parenchyma, it is a living tissue. So why this phloem parenchyma is used? This phloem parenchyma is used for the storage of food. The third thing is the sieve element. This is the sieve element, and the sieve element which is present, it can be sieve tube or sieve cell. The sieve cell which is present, it is present in case of angiosperm, and the sieve tube which is present, it is present in the gymnosperm. So what do you mean by angiosperm? Angiosperm means the flowering plants, and the gymnosperms are the plants which can produce the seeds. Okay, so what is the work of the sieve element? The sieve element which is present, it is a main part where the phloem is being translocated. Okay, and the sieve element which is present, this is also a dead tissue. When it is dead, it is dead when it is in the mature form. Why? Because in the normal condition or during the initial phase, it is having the nucleus, but when it is getting mature, the nucleus has been disintegrated. That is the reason the sieve element is considered as a dead tissue. So as it is a main element for the transfer of the phloem, it requires some nutrients, it requires some of the hormones on the nutrients for mechanical support. It requires something. So this mechanical support is being provided by another part, that is the companion shell. So what is the work of this companion shell? The work of this companion shell is that it maintains sieve element. How? Because it provides nourishment to the sieve element. Okay, this companion shell which is present, it helps in the nourishment of the sieve element because it is transferring the phloem through it, so it requires maintenance. So that is the reason this companion shell is responsible for protecting the sieve element. Okay, here we can see there are different parts, like here these are the sieve tubes. The sieve cells, when it continuously, they are arranged, they are in the form of a cylindrical shape, and when they are arranged, it form a sieve tube through which the phloem is being transferred. And this phloem which will be transferred, it can be transferred by apoplast or symplast manner. If it is transferred through the plasmodesmata, that means it is transferring its phloem in the symplast form. What do you mean by symplast or apoplast? Symplast means transfer of the phloem part or transfer of the sucrose in the by using the plasmodesmata. If it is apoplast, it will not cross this plasmodesmata, it will cross this parts. Okay, it will move through this cell membrane. Here we can see that there are sieve tubes or sieve tube elements is present over here. Attached to the sieve element is a companion shell, and this companion shell is having different type of cell organelles, but there are less cell organelles in the sieve tube element. Okay, so here there is a side plate. Here we can see the side plate, so it is having some pores. We can see that this are having some pores, so it will allow in transporting the fluid part or the liquid part or the phloem shaft to transfer from it. It is working as a what? It is working as a transfer system. Okay, now next we will discuss about some of the features of sieve tube element and sieve cells. The sieve tube elements which are present, it is present in the angiosperms. So what is it? When the sieve plates that when the sieve cells are present in continuous form, they form the sieve tubes, and the sieve plate pores are open channels, and within it there are P proteins which are present in dicots and many monocots. So what is the work of this P proteins? This P proteins will help for the maintenance. If there is some of the damage, then this P protein will help in repairing this damage. Okay, companion cells are source of ATP and perhaps other compounds, and in some spaces are transfer cell or intermediary shell. So what happens, there are these companion shells which are present. They are of different types according to the number of plasmodesmata present in it. This companion shell are of different types. Like this companion shell can be ordinary companion shell, it can be transfer companion shell. Like for example, the first is the ordinary companion shell. Ordinary companion cell. So what happens in case of ordinary companion shell? There are few or no plasmodesmata. Okay, when it is transfer companion shell, there is lack of plasmodesmata, but few plasmodesmata are present. Next is the intermediary companion shell. In this case, there are plenty of plasmodesmata. That means from here, through the process of simplest way, the phloem can be transferred. Okay, now let us see how the phloem is being transferred from the source to the sink. So what happens in this case is this is a place where the phloem is being synthesized or the food is being synthesized. So this food should be transferred to different parts of the plant. So for that we can see here that from the source it is being transferred into the companion shell by the process of osmosis, because here the amount of solutes or amount of the food will be more, so it is crossing this part and it is being transferred into companion shell. And in this companion shell, here we can see that there is simplest, through the simplest or apoplast, it will be transferred into sieve element. Here there is, along with the solute, there is also water present in it. So here it will be transferred through the process of apoplast. Now here what happens, here the amount of solutes which will be present in the sieve element, it will be more. Here there is sieve element, here we can see that here there is sieve element, and here there is xylem vessel. So what is the xylem vessel? Xylem vessel is a part from where the water will be transferred to different parts. So here as this portion is having high amount of solute, that means here the endosmosis can take place. That means it has the ability to take water from the nearby cell, and the nearby cell is a xylem over here. So the water will be transferred from xylem to the sieve element. Okay, so now there is mixture of solute and solvent over here. So here that will be through the process of diffusion or due to the gravitational force, it will be transferred to lower parts. When it is transferring into the lower parts, we can see here through the process, through this pores, it will be transferred here. Through the pressure driven bulk flow, the solutes and solvents will be transferred to this part. Okay, now here what happens, that there is a sink which is present over here. This was a source cell which was present, and from the source cell, this source cell is having high amount of solute with it. This source cell will help in the transfer of the solute into the companion shell, and from the companion shell it will be transferred into the sieve element to the process of symplast. And as it is having high amount of solute with it, from the nearby shell, that is a xylem vesicle, it will help in end osmosis. That means water will be taken up here. And now through the help of bulk flow or pressure flow, here now it will be transferred to different parts. Now this is the reason where the sink is present. Okay, so here what happens, here the amount of solutes is more and here the solutes are less. So through the process of diffusion, or we can tell that, or through the process of diffusion, by using the symplast, it can be transferred to the sucrose which is present. It will be transferred first to the companion shell, and from this companion shell it will be transferred into the sink. So when these solutes are being transferred, so here there will be more solvent or H2O will be more in this region. In the phloem sieve element, the water will be more. So now through the process of osmosis, this water will be transferred to the xylem vessel. Okay, this is a part where the phloem loading was taking place, and this is the part where the phloem unloading is taking place. That means this is being transferred into the sink. The phloems, or the amount of sucrose, is being transferred, or it is being unloaded into the sink. And this process is a active process because it is utilizing ATP during the process of apoplast. Okay, so now through the help of another diagram also we will understand this. So from here, this is the mesophyll cell, this is the mesophyll cell from where the sugar is being produced, and here it is being transferred. Here this is also the mesophyll cell, and at first it is being transferred into the bundled sheath shell, then it is transferred into phloem parenchyma shell, then from this phloem parenchyma it is being transferred into the companion cell, and from this companion shell it is being transferred into the sieve element where the phloem unloading or loading or unloading is taking place. Okay, so in this case we have seen directly this is the source, and from the source it is being transferred into the companion shell, and this is the detailed diagram. So from this mesophyll cell it is not directly transferred into the companion shell. Before that it is being transferred to bundled sheath cell, then phloem parenchyma, then it is being transferred into the companion shell. When it is transferred into the companion shell, it is being done in two pathways, like one is the apoplastic pathway, next one is the symplastic pathway. What do you mean by symplastic pathway? This is by symplastic pathway. That means it is this sucrose which is present, this sugar which is present, it is being transferred to this plasmodesmata. Okay, and this is the apoplast, which is the active loading. Apoplast means it will be transferred from this cytoplasm. Okay, it will not cross, it is not having the plasmodesmata, it will transfer through the walls. Okay, and at last it will use a symplast. It will not directly use from this region, and at last it will use the symplast and go to the sieve element from where the loading or the transfer of this phloem is being taking place. Okay, now what next? Next is what happens when the sucrose, the sucrose which is first transferred in bundled sheath cell, then intermediary cell, then next is the sieve element. So what happens is glucose which is present, it combines with fructose and it forms a sucrose. When it is being transferred into the intermediary shell through the plasmodesmata for the backflow of the sucrose, now here another one thing, that is a galactose is being added over here. So as this is a big molecule as compared to this simplest or it is big to cross this plasmodesmata. So for that, this galactose is being added over here. So now this raffinose which is present, it cannot transfer through this plasmodesmata. So this is done, the addition of galactose in the intermediary shell is done to prevent the backflow of the sucrose into the bundle shaped cell. So here this raffinose which is present, it will be transferred into the sieve element. You can see here that this plasmodesmata which is present in this intermediary shell and sieve element, by connecting this intermediary and sieve element, it is broader than this plasmodesmata which is present in the bundle sheet cell and intermediary shell. Okay, I hope you have understood this. Now next, there are some other transporters which are present, like SUT1, SUT2, SUT4, SUC2. These are some of the carrier proteins which will help in the sucrose H plus symporter. Okay, so this SUT1 which is present, these are SUT1, 2 and 4, these are present in the sieve element, and SUC2 which is present, it is present in the companion shell. And these are some of the spaces in which these carriers are being present. Thank you everyone for watching this video till the end, and if you like this video, please do not forget to subscribe. Thank you.